And perfect. So I share my screen. So I believe you guys, right now you can see my screen, right? There's a paper. And the students can give me a confirmation?
Yeah.
Okay. So this paper is about for the brain, for the neuron tissue. But methodologies, this part is on the virtual part and the drug come to release. So that what like as an example first. And here's like, that's like my collaboration research. And right now it's like Yawen Ye, and he's a neurosurgeon. And at the time it's like PhD students in University of Manitoba. So here's like what we would do is like to using nanotechnology. So nanotechnology to encapsulate a neuron stem cell, a neuron cell, or neurons, neuron cells. And here's like multiple ways, like we would like start, we put like give a cell, a neural stem cell or neural cell coating with a nanofilm. And this one is a cell for examples. And there is nuclear. And to do what? We do some nano coating onto the cell. So we just like to do this like to give a close is to the cell, to give a protection, to regulate cell differentiation. That's possible for future cell delivery to the brain. So what we do, we want this closing could be very dense, density is high or low density. So the porosity, permeability of this nano gel, because it's nano closing on cell, to protect against the cell apoptosis, so means die. So here's like we have first one abstract, and so this abstract is to do what? So like this one, cell capsulation using macro gel or nano gel, means that the gel could be a nano level or macro level. For the cell surface engineering to mimic niche, means microenvironment. Of the cell living. Organoids, organoids means like multiple cells to make a mimic of organs or tissues. And this one purpose like to establish the initial microenvironment can season to regulate cell tissue for controllable development. That's like it's very important. We can use it. We just not like put a bigger tissue or bigger volume. We just use a nano closing to tune a single cell. That's interesting. Like so excuse me. So it's a layer by layer nanogel coating. And we say the bottom up is from cell level to tissue. Cell to tissue. From bottom level to the high level to make the artificial tissue and organs. And so here's like we report a nanogel encapsulation on individual neuron cell, like this one, neuron cell, to start what? To start the nanogel, or you are matrix or scaffold, but this scaffold only for single cell. To do what? To study the permeability, the porosity. And the ones to can tune the cell, apoptosis. So the why, so that then this one is general objective, right? And then materials we used is gelatin hyaluronic acid. Why? Because this one is positive charge, this one is negative charge. We can negative, positive for self-assembly onto the cell. Because cell is negative, cell, we know the cell film is negative charge. So we can coat with gelatin, is positive charge. Then again, negative charge. That's the arginate. That's the HA, hyaluronic acid, arginate, both possible. PC12 cell. This cell is a neuron cell. Neuronal cell. And the permeability we can, by, for example, the concentration of layer thickness and pH value. So that we know the permeability of tumor, we to evaluate TNF alpha is a kind of regulators for the, and by tuning encapsulating parameter for blocking interaction with TNF receptor 1. So in that case, to start cell apoptosis or cell death is to inhibit this procedure. So this one is like kind of a short summary. In short, nanogel can deliver control permeability to different molecules, and we can use this one to screen effects on TNF alpha for protection, to protect the neuronal cell PC12 cell. And the significance, we know that's, technique holds great potential, for the stem cell or neuronal cell implantation. So this one is like introduction. We already, have in your proposal, have pretty good background information. That's the background information. That's more about the general topic is about cell transplantation therapy, have been regarded as a helpful therapeutic strategy for to treat nervous system disorders such as stroke. So that's for could be like using the mini and brain research, and we can inject the cell. But in that cell, we know if directly inject, most cell will die overnight. So general outcome of this treatment is not successful. So critical things is a low survival rate of transplanted cell. You guys remember? Cell, we have stem cell, but if only cell inject, normally the cell will quickly diffuse into other place. Another ones, the cell will die overnight around, for examples, over one night, 70 to 80% of cell will die. Okay, so mostly caused by cell apoptosis. So following cell grafting, so we can bind into the membrane to take effects. So for examples, so there's a TNF alpha can interact with this receptor. This one, the receptor we know in electrophile, it's on the kidney cell membrane to activate the downstream cascade. So it's one's leading to the cell die. So apoptosis is kind of format. It can like a type of the cell to die. So unfortunately, currently no specific way to deal with. So that's like mention like some concepts. Okay, we know this was is cell transportation is very important. However, cell injected to the brain or to the body they will quickly die. One pretty, kind of high frequency is like cell apoptosis. So why they will do? Because the TNF alpha. So here's like, therefore, their rapid developing materials science began to draw research attention in the recent years. So that's like materials scientists have done lots of work to address those concerns. So one way is cell surface engineering. Then we mentioned the cell tissue engineering, cell surface engineering. And the basic things like for examples, like is cell encapsulation and to create a microenvironment. So this microenvironment can promote cell survival and living and differentiation. So that is cell encapsulation. So one way is like we go further. One way is layer by layer microgel. Dual contributes a lot to cell surface engineering or cell encapsulation. So then they also start to define what is LBL. LBL is layer by layer based on the positive negative charge, electrical interaction. So that's the kind of LBL general introduction. So having a wide applied drug delivery and cell protection. Then again mentioned, there are some other issues. So permeability is LBL. We can definitely, we need let this close, have some specific porosity. That'd be good, like for examples, if a close, like we can control the permeability or porosity of textile. So let the humidity or sometimes the air can be, make some what? Yes, So this one is closed. So the humidity or come in and out. So the oxygen, H2O, that's the body. So let's like feel comfortable. So them things for the LBL. So we need to know the permeability. So that's like the ways they work out. So that's the ones to mention, to make a statement, to claim why permeability is so important. And with a comprehensive understanding permeability, so they mention this one, so you this one to do, TNF alpha, because that's the permeability has a huge impact on the TNF alpha. So we want to use LBL to protect from cell, apoptosis. So in this study, we first then make a short like this one. After introduction, you make like a short study what we will do. We first establish nanogel encapsulation, then use the gelatin etchant, the permeability encapsulation by different parameters to the FD with different molecular weight. So that's like we have kind of fusion or diffusion parameters. We can use this one, right? It's a FITC dextran. So that's like a materials method we will use. That's dextran. We know it's a molecule. We can control the different dextran because that's the polymer. We know have different size of polymer. And with FITC, it's a fluorescence chemical. So diffuse in or diffuse out, the fluorescence will make a change. And meanwhile, we have different size of dextran. So we know the porosity. So first one, a lot of students are still confused about this part, okay? Materials and the method. So what materials use? So first one, we use, FRTC dextron because this is a molecule, a modeling molecule. We now use this one, yeah, for examples like we put this one is FID. We want to go to like diffuse this one. This one is an LBL nano gel. Then this area is a cell. Okay, so that is the purpose. And we have different size of dextron FD. And we can compare like the LBL, the density, concentration, pH value. So that's the method. One is FITC, and we have some other things like boron, serum, abyssum, we will talk, yeah. And lots of materials used, the solvents, PVD, PBS, and the chest, that's like to stand the nuclear, and some fluorescence FITC, that's for fluorescence. We have the materials, yeah, hyaluronic acid, HA, TN alpha, yeah, that's like that's antibody, right? We use, and the protein G, that's like a lot of chemicals. That's you can follow this way. Yeah, that's one to start the neuron cell, and some tonic kit that's all started in the PC12 neuron cells. So that is the materials part. Then this one start is the methodology part. Nanogel, that's most important. Nanogel. So as previously described, that's like, that's his first paper, LBO, with 2 million neuron cell lines, PC12 cells, first incubated in 1 milliliter gelatin solution. And with different concentration. First one stay there for 10 minutes and the gentle shaking because this gelatin is positive charged. We know cell is negative charged. So negative charged cell, each cell will let the gelatin, a positive charge, deposit onto the cell surface. So that is mechanism. Cell was washed then after the gelatin deposit onto the surface. Then we centrifuge. We know the centrifuge, and then we get to the cell will be form a pellet on will drop onto the bottom of the centrifuge tube. Then washed with DPBS. So that's the buffer. Yeah, and the centrifuge again. So we remove the upper part. We only collect the cell. That's the pellet in the bottom, like this one.
And that look
so
is high detail
at the beginning
shallow depth of field
and see
all the time. So literally anytime you have a close-up
any question,
you can count on the background
that's
being
the
blurred out of focus.
tube.
None of these
At the
shots are
beginning,
portrait mode. This is just straight
cell,
snapping photos. It absolutely epitomizes that big sensor look in a smartphone.
already
but guess
like
what? That also means
coated with
if you
gelatin,
have more
then
subjects
centrifuge.
Like I do, you're going to start to see
so what we can
around
get
the outsides
after centrifuge. So all cells be here. So we, and this part, we just get this part out. That's the cell. Then for another one is for HA. Hyaluronic acid, because right now the cell coated with gelatin. So with positive charge, then negative charge HA go. Then for the multiple layers, the cell, Oh. That's with gelatin. then with HA, you can layer by layer, and again, generous like they will make another gelatin, and you can put another hyaluronic acid. Okay, so layer by layer depends how many layer, so you can start what will happen to the nuclear to the cell. That's when nuclear. Okay. So that's LB L layer by layer self assembly. Okay. And when we get this one, we definitely need a TEM. If you have, we know the midterm, right? Nano level. So what do we do? TEM is one. If you mention SM, that's wrong. Okay, TM is okay. TM and some like AFM, atomic force microscopy also both work. TM was as previously that's from reference. So untreated PC12 cells and the PC12 cells encapsulated with like 8 layers. That's always we made 8 gelatin HA layers were first fixed. That's the methodology. A glutaride, that's like the hide for 1 hour. Then we put like a buffer and in the overnight, then that's the TM methodology. For the post fixation, then make a slice as atrazine film for TM. So then were dehydrated with gradual alcohol embedded with epoxy resin. So then dry 60 degrees for 22 hours, then using a macrotome to make a chip, make a tracing film, and then mounting that on the mesh for TM. And another one's atomic force microscopy. That's also for nano level cells seated on a cover slide. Then using microscope scanning. And so we on an area around 25 micrometers on the surface were bigger, and the preparation of fluorescing labeled gelatin. So for the preparation of RTC labeled gelatin, 20 milligram gelatin was dissolved with the buffer, and FITC was inside. So that's one gelatin with FITC with fluorescence. Then FD permeability investigation, the cell were encapsulated with LBO membrane. FD with different molecules, then incubated with cell in medium with pH, different pH, 30 minutes. So we see how many molecules have been permeate inside, go inside. And this one is like immunofluorescence. So this is all about in vitro study. Immunofluorescence. So PC12 was seated on cover slide. Then take a look at for the immunofluorescence study. So for this one, the cell nuclear we're staining test. That's we can stain the cell and cell body with different color with the nuclear. So from nuclear, we know, we can count the cell number. And primary antibody we use KR67. That's for to test the migration or the cell or like the cell toxicity. And the PLA, the TNF alpha, was incubated with cell and blocking. That's the methodology. Okay. And the Western blot, so that's the method to take a look at the interesting plotting expression. So we can use this way to measure TNF alpha and their concentration changes. So for examples like that's the methodology, yeah, and we have a statistic, then the results. So first results is the cell compatibility study. We want to check cell living, death, or keep that cell's property. For example, the stem cell, for example, right? So we know like PC 12 cells extract from rat, yeah, adrenal medulla used since the analogy as the neuronal cell model. And with gelatin, make some results. So you will see like cell viability test, that's important. In capsule with different parameters, we detect with her chest PR staining. And the red, yeah, means dye, for example, it's like after being coated with like different layers, then we check like this one, that means style cell, okay? That means the cell dye. So we need a majority 90% or 99% is no red, okay? That means they're okay, yeah, they're good, yeah, okay? That means like the, yeah, her chest PI staining to detect the cell die or, yeah, or die or living. And so that's like expected results. And we can counting cells. So from this one, yeah, proliferation of cell, untreated P cell, that's as a compression, and encapsulated cells. So KR67 staining, untreated, and cell capsulated. Yeah, we just like quantify the KR67 numbers for each group, then measure the cell number increase and decrease. And characterization, yeah, characterization, for example, is like single cell encapsulation PC12 cells. Yeah, single cell, yeah, means like you see a cell, and yeah, that's the membranes here. Yes, if no, like this one, FITC or fluorescence. So inside the cell, outside, we know the gelatin with FITC. So we can check. Also, we can from TM to check the surface layer. If you see this layer is a nano layer, that means to verify, using TM to verify the cell coated or non-coated. Again, we use FM for further confirmation. We can get to the results. Then we mention like, layer by layer to talk about FD, diffuse into or diffuse out. Yeah, if like the mesh is very dense, so the bigger molecules cannot go inside, and a small molecule can go inside. That's the FD. If like the mesh is pretty loose, so we big, small, both can go inside. So we can from fluorescence, we can know what's happening, right? Yeah, that's the control without any barrier. So lot of blue, green inside. That means no, and we see like 8 layers, and we can check, yeah, what's happening inside. Again, that's kind of a permeability research. And then we studied even further for cell level. So that's like stop here, cell counting. And that's because that's the in vitro study. Then in vivo, again, that's my previous PhD students. Right now is, yeah, moved to Harvard. Yeah, got MIT Harvard's offer. Right now is at UCLA. Yeah, last year he moved to there. And that's research is the hydrogel. Gel acting to treat disease. So, and this one will be a in vivo model. So you will see materials method parts. We will check, yeah, that's also gel and physical property of the hydrogel, because I know some students working on the ball ink. So we need to measure some swelling test because yeah, the gel will absorb water. So how much water can absorb with time? So we should know. So measure the swelling behavior. NMR, that's we know, that's to verify the chemical structure of your hydrogel or ball ink. This NMR and the in vitro degradation study. So you need to know in the water, in the PBS buffer, with time, the quality, the weight change, okay, weight change. We know, with time, for example, the degradation rate. Rheology testing, that's how hydrogel, because hydrogel biomaterials basically is a viscoelastic materials. So we need to do kind of rheological hydrogel. Then cell culture work, right? What kind of cell culture? How do you cell culture? MTTS. MTTS for what? Is to test the cell toxicity, cytotoxic to cell or cell swab ratio. That's the ones very important to test cell biocompatibility in vitro. And the drug treatment, we know like the acting would be we can get. And again, contrast staining, you guys remember, right? It's very similar to last paper to take a look at the cell, to look at how many cells die, or measure the in vivo release. We put the drug is acting B in the hydrogel to take a look at is the gel, the release efficiency or release rate. And the in vivo experiments, the animal, that's one. A lot of students in this term, we need to conduct some in vivo. So animal. So what animal? Is male mice. So we generally use male, not the female mice. Okay. So that's very stable for research. And if right now, if you submit a paper, research paper to publication, they will ask you why choose male rice for study or why choose female mouse as your study model. We need to expand. And meanwhile, we need to have ethical approval. That's like, with ethical protocol. For example, follow NIH protocol. And so how many groups you will make, and we will make a Parkinson disease model for research, and conduct how many days for your research. And so immunohistochemistry, that's all students need to focus on this. Need to have this part, because that's like to study what change in your disease model, and we take out the tissue and to do some immunochemistry, use antibody. And so here's like some results. First one, how to make the materials, and the morphology. That's the SEM image, because it's a macropores. That's for hydrogel, not nanogel. And with NMR, right? That's HMR to detect the chemical structure and the degradation date and the swelling. So the results we know, like this one. That's a rheology testing. That's the gel, because that's the temperature sensitive gel. In the room temperature, it's clean. It's a clear picture. In the 37, they make a cross-linking, make the gel, so it's non-transparent. And that's one. it's the needle, injected into the body, into the brain, because that's for Parkinson's disease model. That's for Parkinson's disease. So we first create a Parkinson's disease, the mouse, and use a atrazine needle injected to the brain, right? So we know lots of areas, and for that's all related to, we say what? Immunohistochemistry research. So that is my previous research. I also give you some examples like how to write the material method and the expected results. Any question? Any question? So please send me your slides. I could give you comments before. Don't forget, again, I want to go back to that part. 5 minutes, your team name, contribution, each contribution proposal, contribution object, background, material method, results or significance, conclusion, and some reference. Any questions? If no question, our class is over. And if you want to book a meeting with me, please send me email. And if you could send me your slides, I could give you comments. And so before the class, before the Wednesday, you need before the class, send me your slides. I need to take a look at your slides first, and then because right now in the Zoom meeting, so I will take a look at the slides first, before your final. So that's the please right now you can send me right now the slide. I could give a comments, and before the class, you please send me the final slides. That's for your evaluation. Thank you.
Final exams this Friday, right?
No. Final presentation is Wednesday, right? And the final examination is like a release. That's like for 4 days. Like your midterm. So it's different.
When would you release it?
Release is a Friday.
Okay, thanks.
Okay, good. So see you guys.